morning, everyone. Thank you for joining me today for the presentation of Dyadic's protein expression platform with a focus on our C1 technology. I'm Joe Hazelton, Chief Business Officer for Dyadic. Before we get started, anything I do say may be forward-looking, so Dyadic takes no responsibility or obligation for forward-looking statements. Oh, there we go. At Dyadic, our mission is to make proteins for world health to meet the rising demand and unmet need for therapeutic and other proteins, to address the current and future health threats and nutritional deficits. We strive to meet the demand through applications and adoption of our highly productive and scalable microbial platforms to turn DNA into commercially relevant and affordable enzymes and proteins. We've invested significant time, energy, and resources in developing our filamentous fungal microbial platforms to produce proteins equivalent to CHO and other traditional platforms, but in higher yields, less time, and lower costs. So to take you through the history of Dyadic, today we have two primary production platforms: the C1 platform for human and animal health pharmaceutical applications, and the Dapibus platform for non-pharmaceutical applications. At the beginning, Dyadic targeted the industrial enzyme market for textile manufacturing, but over the last 30 years of development, we've learned how to genetically engineer microbial cells for broad applications beyond textiles. To do so, we optimized the molecular toolkit for broad development across diverse industries, then developed an enzyme library, developed an enzyme library and achieved generally recognized as safe or GRAS status by the FDA. And now we've established global partnerships with industry-leading organizations like BASF, Shell Oil, Abengoa. After selling our industrial business to DuPont in 2015, we embarked on the development of the C1 platform for the rapid production of large quantities of antigens and therapeutic proteins to lower the biomanufacturing costs of animal and human pharmaceutical products. With the C1 platform currently in phase 1 studies that so far have shown to be safe and well tolerated, we're currently engaged in several partnerships with leading human and animal health pharmaceutical companies with the C1 platform, and we are seeing accelerated adoption of our Dapibus platform for non-pharmaceutical applications, such as the development and licensing agreement we announced yesterday for an animal-free dairy enzyme. All cell lines in biomanufacturing must meet certain requirements in order to be commercialized. The platforms must be able to properly clone, express, correctly modified, and folded functional proteins. But what makes a cell line truly unique or an ideal choice for an expression is also one that's highly productive, fast, and cost-effective. This is why we feel the C1 protein production platform is uniquely suited to meet the rising demand for therapeutic and other proteins. C1 can produce standard or complex proteins that have been demonstrated to be comparable in function and quality to those produced in other cell lines like CHO. C1 has now entered human clinical trials with favorable preliminary safety data, but has also the capability to produce larger quantities of proteins at lower costs and in less time than traditional platforms in use today. The COVID pandemic has clearly identified critical shortfalls and bottlenecks to providing rapid, adaptable, and sustainable preventative and therapeutic antigens and antibodies to service a global population. A global solution must be affordable and available to all regions in order to prevent or combat future pandemics. Platforms must be agnostic to not just route of administration, but also agnostic to infrastructure, economics, and distribution capability to all regions. C1 has produced a record 10 grams per liter of a livestock antigen with consistent production in the single-digit grams per liter for human antigens, and we've produced a monoclonal antibody at over 20 grams per liter while remaining cost-effective for developed as well as emerging countries. We've structured our business development in 3 core verticals. Human health and animal health are the cornerstones of our efforts, with C1 supporting the pharmaceutical applications in these verticals. The third vertical of alternative proteins is supported by the Dapibus platform for all non-pharma applications. This segment the alternative protein segment, represents more near-term potential for Dyadic revenues given the shorter development times for commercialization. C1 is an extremely versatile platform with broad applicability across pharmaceutical development and supply chains. C1 doesn't just have high yield and low cost antigens and antibodies. It makes complex proteins like ferritin nanoparticles, trimers, full length MABs, and multispecifics with the quality and performance of 1 million cell lines, but in larger quantities, in less time, and more affordably. Additionally, C1 is capable of producing pharmaceutical development and supply chain products like diagnostic antigens and antibodies, catalytic enzymes like DNase I, as well as recombinant human and bovine serum albumin. Despite the robust versatility of the C1 platform, its manufacturing process is relatively simple and straightforward due to its industrial heritage. The C1 fed batch production process can be implemented in almost every region of the world, as it's cost effective due to the high productivity and use of fully defined low-cost media. It also remains adaptable to a wide range of growth conditions and temperatures, and has a short 4 to 7 day fermentation process. Additionally, due to C1's novel morphology, which changes from a typical viscous filamentous growth during fermentation to a fragmented mycelial phenotype that grows under very low viscosity, this can be run at extremely high levels, all the way up to 500,000 liters. Not that you'd need that in human pharmaceuticals. These proteins are typically secreted, and we can reach a stable cell line in approximately 6 weeks with enough material for preclinical studies. C1 is a non-sporulating filamentous fungal strain, therefore it can be run in cGMP facilities, and we've also run our own cGMP manufacturing run for our COVID vaccine. Also, we learned from COVID-19, not only is the productivity of cell lines important, speed is critically important as well. The short fermentation times and lack of DSP viral or endotoxin removal means C1 cannot just produce more proteins in traditional cell lines like CHO and baculovirus, it can produce them more quickly as well. This means more preventative and therapeutic proteins are available in less time, which is critical for pandemic or regionalized health threats. We spent the last 20 years developing the proprietary and patented C1 protein production platform to produce stable, high-quality therapeutic proteins. Dyadic has the technology and experience to apply our learnings to develop targeted project plans based on the genetic tools we've developed, such as multiple integration techniques, use of different promoters, spiking library assays, folding chaperone library, as well as fermentation and media optimization. Combined with our industrial heritage and scale-up expertise, this enhances our ability to rapidly produce larger quantities of low-cost therapeutic proteins. The pandemic has highlighted the shortcomings of the current production methods in use today. While mRNA has proven to be a rapid production system, there is a need and a desire for a recombinant protein solution that can meet the 100-day identification to inoculation challenge. C1 can currently generate stable cell lines to produce enough quantity for preclinical testing in as little as 6 weeks, but we believe we can reduce that even further with additional development and regulatory considerations. We believe this may enable the development of a recombinant protein vaccine within the 100-day timeline. We've also developed a library of over 50 proteases through a costly and time-consuming systematic deletion process to improve the stability of the C1 host for monoclonal and other antibodies. This ultimately improves the quality of the C1 host and quality of the monoclonal antibodies expressed in C1. We've also made substantial strides in our glycoengineering capability, and we utilized nivolumab as our initial monoclonal antibody for expression due to its market dominance as Opdivo, that's Bristol Myers Squibb's $8 billion drug. The native glycosylation pattern from C1 has been humanized to produce CHO glycans and has nearly all human-like N-glycans, over 99%. The remaining percentage is MAN3 species, and for site occupancy, our relative response is close to the values of the Opdivo reference standard. The tools and skills we've developed for commercialization of the C1 platform have enhanced our capability to produce newer and more complex proteins. Just as our ability to produce complex proteins is also the retention of key benefits of the C1 platform, which are high yield and low cost. We've produced VLPs, nanoparticle antigens at up to 3 grams per liter, in addition to high productivity of trimers and proteins with an AMHC2 targeting capability. C1 can make complex proteins at high yields and lower costs, resulting in broader applicability in human and animal pharmaceuticals. We believe the C1 protein production platform can enable the rapid and adaptable commercial manufacture of vaccines and therapeutic proteins. Here you can see examples of the breadth of antigens, monoclonal antibodies, and glycoproteins expressed in the C1 system. In addition to that broad capability of expressing molecules, here you can see the productivity, the high productivity that's been reached across the spectrum of antigens and antibodies, demonstrating that the productivity of the C1 cells spans the different biomolecules and disease areas being targeted. Soon we'll be able to update this slide with other proteins and enzyme catalysts that are crucial to the development and supply chain management. So to summarize, with Dyadic's microbial platforms, you simply have the potential to get more for less faster. C1 can produce therapeutic proteins and antigens that are comparable to CHO or baculovirus, if not better in quality, stability, and human glycan structure. C1 also has much lower upstream costs, higher productivity potential, and faster development times, which translates into more preventative and therapeutic products that are efficacious and affordable for all geographies, which in turn provides the potential to help solve pandemic and other health threats. Thanks for your time today, and be sure to follow Dyadic in our future journey to deliver proteins for world health.